Well, thank you to the organizers for choosing my abstract for a talk. It's very prescient to be talking about the cannabis pangenome. So the image that you see on the slide was actually created by my lab manager as we embarked on this pangenome extravaganza. And what you see here is actually the civilizations that have risen and fallen during the period of time that cannabis has been around. So cannabis will be around regardless of what is going on in the world around us. And which inspires me, and this is where I started my journey with cannabis. When I was younger and reading that magazine that used to have nice pictures of plants, I saw this and was inspired by the fact that this plant can produce, by dry weight, so much material in these trichomes, the furry part of the plant. And it inspired me that there was something that I needed to know about how this plant works. So that's where my journey began with this. But it wasn't actually really until long read sequencing came around. And I'm not going to talk about sequencing in this talk. I'm just going to talk about genomes because now the technology is caught up to a place where we can pretty much sequence most plant genomes, which are the most complex genomes that we know of. So this was my first foray into the cannabis genome, which was CBDRX, which was an inbred line, high CBD. And the nice thing about it was that it was inbred, so when we sequenced it, we actually got fairly good resolution of the genome, which allowed us to do something that I had been wanting to do since talking to Kevin back in like 2011, which was understanding the terminal synthase pathway or the gene cluster that was responsible for it. So what I'm showing you here is the chromosomes of cannabis, and we identified the synthase locus on chromosome 7. But moreover, what we found is that it actually was nested in this very interesting transposable element cassette, and that each of the synthases was actually nested between some transposable elements, and that these cassettes were in standard formation. So the main genome from this paper was the CBRX, but we added in a couple other genomes to see was there something interesting or different about these cassettes. So I'll come back to this, but for me, this is what science is about. So when we first got this sequencing back, and I was able to assemble the genome and look at this for the first time, it really changed the way that I thought about the plant. But it also, like, in this paper, we showed that potency wasn't responsible, or the synthases weren't responsible for potency. And this really opened up a lot of other questions. And hence, we needed the pangenome. So this collaboration started with Oregon CBD, who actually generated most of the genomes that I'm going to show you today. So about 75% of the genomes had been produced by Oregon CBD to look at their breeding population. And so they contributed all these genomes, and this is the pedigree of the genomes in the pangenome. So if you're interested in seeing how alleles are shared and go through populations under selection, this is a really interesting data set, and we've only scratched the surface of it. Two very important people that led this work are Ryan Lynch, who is actually a long-time cannabis breeder, and then Lillian Paget-Cobb, who was coming from the hop background into the cannabis field. So underpinning the pangenome is a high-quality genome that we generated, which we call EH23. Which is actually a cross between a type 3 and a type 1, so it's a type 2 plant. And we utilized this to do several things during the pangenome construction, but what we needed really was a reference genome that solved some of the previous problems. And some of the previous problems that we had with the other genomes, even CBDRX, were regions that we're really interested in. So this is basically just a dot plot of the genome. So a dot plot is when you take, in this case, the two haplotypes of the genome and you compare them together. So a haplotype is basically one set of the genome, which usually is the only thing that we could look at in genomics, but over the last 3 or 4 years, like I said, long-read sequencing has dramatically changed what we can actually see. So cannabis is diploid, so we can see both of the chromosomes, both haplotypes. And what this exposed is that this region where the synthases are was highly variable between type 1s and type 3s. It also allowed us to resolve some of the other issues with the genome. So once again, these are dot plots, and now it's one haplotype against CBDRX, and you can see that like the little dots are all over the place. That's because in CBDRX, we were unable to properly assemble all of the centromeres. And centromeres are important. It's very gene-poor regions of the genome, but now we've been able to actually resolve them. And then the other one is another genome on NCBI that was getting pretty close to really high quality, but we were able to fix some of the final issues in the genome. However, we like to look at the genome slightly differently. So Brian Canals from Kelly Vining's lab at Oregon State came up with this way of viewing the chromosomes, which I absolutely love because it actually shows us several aspects of the genome. So the colors are actually the gene density along the chromosomes. And then what I want to point out is if you look over to chromosome 7, you can see the orange triangle, which is that inversion in the genome where the synthases are. Super awesome to see this. There aren't any centromere sequences, so generally plant genomes have very large centromere arrays, which are repeated sequence of usually about 150 to 300 base pairs of the exact same sequence. Very difficult to assemble for plant genomes. Turns out that cannabis doesn't have them. It has these GC changes in the sequence, so very odd for a plant genome. So when we started this, Oregon CBD came to us with many of their genomes, and we had some of our own, and we then went and unified the whole data set. So when you go and look at the pangenome, you're not just looking at a bunch of genomes brought together, but we've unified them in several aspects. And one of the really important aspects of the unification is actually the gene prediction, which if you are someone who looks at genomes and analyzes genomes, you know that genes are actually, well, one of the most important things that you want to look at, but also one of the hardest things to get right at this point in genome research. The other thing that's really interesting about this, and I already mentioned Oregon CBD, and they had their whole pedigree, but we also added in genomes from the USDA germplasm resource. So in other words, these genomes, you can actually order the seed from GRIN and actually study them, and then you have the genomes to back that up. And that was one of the things that I thought was really important for the community so that you could actually access the germplasm and then do the research on the plant itself, which was really one of the limitations that we ran into with the publication of CBRX. I'm not gonna go into how we do this, but this is a representation of a pangenome. This is from our tool called Pancamer, and what you're looking at is a Jacquard matrix, distance matrix, that basically takes all the genomes, full genomes, and fairly rapidly can ask, how are they related? And so what you're seeing here is basically a matrix where the darker color suggests that they're related, and the lighter color is not related. I know you can't read the labels, but I've highlighted a couple things. I always like to point out that white widow, haplotype 1 and haplotype 2, now that we can actually see the male and female chromosomes, you basically can see that they come from distant places, and this is reflected in how interesting this plant actually is and why it's still one of a very utilized plant today. But you can see how the different types of cannabis break down, and there's a great amount of diversity. So I don't have a comparison here, but when we look at the pangenome of other crops, you don't see this type of distance between the different genomes. Another way of viewing this is actually in this plot called gene space. And what I'm showing you here is actually the chromosomes, and the colorful lines actually show the relatedness between the chromosomes. Here I'm also including the X and the Y chromosome, and you can see which portion of the X and Y are actually shared. That's the first line right here. But then the part that I want you to see, and I'm going to highlight this later, is that the white space along the colorful lines, and these are places that the genomes are different. So it turns out that the cannabis genomes are dramatically different. The most diverse structurally of any genomes, any plant genomes that we've looked at to date. This is another way of looking at it. So this is a tool that Kevin Fengler told me about that he uses in his lab at Corteva. And then we codified it into a tool that now anybody can use. It's called Pandots. So if you have a whole bunch of genomes, you can actually bring them together and actually view them. And what I want you to see here, which is really the only part that's you can actually see on the screen, is the places that are different. So places where you get the breaking. Once again, I wanted to highlight there's a lot of structural diversity in cannabis. This is one of the requisite plots for pangenomes. And so imagine that you're sequencing a whole bunch of species. You can model a bunch of cultivars. You can model how many you need to cover the whole genetic diversity of a species. The other way to do this is to sequence a whole bunch and then see how much of the genome have you actually captured. So we call these rarefication plots, pangenome plots. The protein plot, this is we take all the proteins from the pangenome and then we ask if you add one genome more or one proteome more, do you see anything new? And if you get this plateauing of the blue peak, that means that you've pretty much captured all of the proteins in this species. The second plot, which is based on Kamers or the genome itself, you see that it's not plateauing. What that means is that we've only started to scratch the surface in terms of the diversity of the genome, which could be these transposable elements, or it could be other factors that are actually really important. And I'll mention this too later. I actually think that the way to read this is that the genome we haven't even touched yet. And while the proteome, it looks like we've captured a lot of the proteins, how can that be? Well, it turns out that cannabis has diversified by tandem duplication. That means it takes genes that are important, usually in the biosynthetic pathways, and amplifies them. That means that we would not see any new proteins per se, because these amplifications are the same proteins that have been amplified. So there is a lot to still discover, but mostly in these amplified regions, and that's important for later in the story. This also can be seen in the size of the chromosomes. So this is basically a plot just showing the number of genes by the chromosome length. First, I'll point out that the Y chromosome is massive. But there is a lot of variation in these chromosome sizes, and this goes to the features I've already mentioned: structural variation and the tandem duplication of genes. This is another way of seeing this that Ryan Lynch came up with. It's actually looking at the places in the genome where we see either inversions or duplications. And you can see there's very specific places in the genome where we get a lot of these inversions or duplications. I want to point out one on chromosome 1. We get this huge inversion, and it's actually in a very specific part of the population, and it's related to autoflower, or just flowering in general, where these loci have been locked in place. So inversions are actually really important because they become bottlenecks in a breeding population. So it's actually very good to know about these kind of things so that you can work around them. So what's the utility of a pangenome? It's to see all these different features so that when you're making your crosses, you can accelerate your breeding cycles. All right, so going back to the synthases. So what I described earlier was that in our initial CBRX assembly, we saw that the synthases were nested between transposable elements. So basically you have the synthase gene, which is intronless and and there's only one full-length copy in the genome, and it sits in between two fragments of transposable elements. And then those genes and transposable elements are arranged in the genome next to one another in what I call arrays. So I want to know, like, okay, so we have all these genomes. Are all of the synthases the same, or are these arrangements the same across the pangenome? What we found originally in CBRX and then a couple of the other genomes that we looked at is that we didn't see any difference. There were differences between type 1, so the THC clusters, and type 3, the CBD clusters, but there wasn't much variation between the two. So we looked at the entire pangenome, and I don't expect you to read this, but like basically you can summarize all of the synthase clusters across the entire pangenome and then ask, well, how many different versions are there? Turns out that there are very limited versions of these clusters. So in other words, in genomics and genetics, what this means is that this locus has been selected for and pretty much bottlenecked so that we have very few combinations of these types of genes. In terms of, CBD arrays, we only found 2 different ones amongst all the ones that we looked at. There's a little bit more variation in the THC synthase clusters, but really not that much more. So this locus has been highly selected on. This is in contrast to the entire pangenome, which is very variable. And the other thing that I want to point out is that even though these are conserved, they also show lots of structural variation. There's structural variation, but they all show up at the same place. So what I'm showing you here is we've normalized the genomes by using standard proteins, and then so basically we can look at the positioning, the relative positioning of the synthases. On this side, I'm showing you the whole chromosome and then a zoomed in version just of where the synthases are. And what you can see is that basically there's a very standard organization across the genomes, which once again, I showed you previously, there's structural variations around these, but they basically stayed in the relative position. This is using that PanDots program. And what you can see is that there's so much variation, so many structural rearrangements here. Why would you have so many structural rearrangements but maintain the same position? It's because it creates breeding drag. So basically because they've been selected on over and over and over again, you create these islands basically of these genes. So there's a lot to do here to untangle this, but this suggests that we basically, in the germplasm we're looking at, we have really maxed out on the synthase cluster, and this is not where the variation is in the pathway. So where is the variation? Well, this got us thinking about this wonderful biochemical factory. So I just love looking at these trichomes. This is where all the cannabinoids and terpenes are made that we're interested in. Obviously, the plant also makes cannabinoids and terpenes in other places. But it's these biochemical factories that inspired us to think more broadly, is there a part of the pathway we haven't actually looked at more closely? And one of the things that I'm very interested in are all of the different cannabinoids. Minor cannabinoids, or the cannabinoids that we've all been talking about. They're actually, the presentations yesterday in the practicum were really great in terms of when I talk to people about cannabis, people are really unaware of the endocannabinoid system, which is not surprising, I suppose. But when you start to dig into it, it's really fascinating how there's all these other cannabinoids that are actually having very interesting effects. So one of the things that, the reason why we had this pedigree of plants is that Oregon CBD wanted to move the propyl or varin trait into their breeding populations. So it turns out that HO40, part of the EH23 cross, is high variant. And so in this cross, they made 288 individuals that we could use for mapping of this trait. This is basically a QTL study, but because of the diversity of cannabis, we actually had to analyze it using genome-wide association methods. And we identified two main loci, BKR, which had already been published, and then ALT4, which had been hypothesized as part of this pathway. And we've really just focused on these. What we did identify two other loci that we haven't actually really focused on at this point because they were more cryptic and we were unsure what the loci was actually is because they're located in these big dense islands of transposable elements. All right, so what did we find? Well, the great thing about having a pangenome is that then you can look at your entire population and say what's going on in terms of this gene. Now, it turns out that HO40 has this 2 base pair deletion, which actually knocks out the activity of BKR. So we confirmed what was already shown, but you can also see that there are other variations in this region. So what I mentioned earlier is that these structural variations lock in certain genotypes. Now, in our feral hemp, in our European hemp, our Asian hemp, and also in some African varieties, what we're finding is this dual inverted repeat right at BKR. So one way of actually locking in a trait is actually creating a structural variation. When you actually make that cross, you cannot actually change that structure. So in other words, you get BKR every time you cross it that way. So we haven't started to explore this, but it suggests that there's a lot of variations still to be tapped in this locus. So the other thing that you can do with a pangenome is you can ask, okay, what are the different alleles underneath all of these loci? So one of the things that we did was, I showed you the pedigree before, and that pedigree was actually to move the variant trait into their breeding population. And so what we did is we utilized this pangenome tool that I mentioned previously that allows us to actually take all the genomes and identify regions that are shared. So it's sort of like we can trace alleles through that pedigree. And what I'm showing you is that when we trace that pedigree, we can find recombination events, and that's shown by the dotted lines here. So every generation, you get 1 to 2 crossovers per chromosome. And we can actually follow those crossovers, which allows us in a separate way, different from QTL mapping, to pinpoint the genes associated with a trait. Some people call this integration. I like to call it allele tracking. And what this showed us is that the HO40 has this ALT4 allele in the population across the entire pangenome. What I'm also showing you, though, is something that I don't show very often, but it's very popular in the pangenome realm, which is a graph pangenome. And we find that they're very useful for localities. This is this right here. This is the graph pangenome just for the region that has ALT4. Now, this is sort of like, and maybe some of you have seen these, like systems biology plots. You really can't make any sense of them by looking at them like this because they don't really make any sense, but they make pretty pictures. But the take home is that this region with ALT4 is super complex. And the reason it's super complex is that these genes have been tandemly duplicated. So going back to what I told you before, cannabis has expanded its chromosomes by tandemly duplicating important biochemical pathways. ALT4 is one of them. Now, in this study, we were able to locate this trait to this part of the chromosome, the top of chromosome 7. So in other words, it's also linked to the synthases, depending on where your recombination is. We actually had to look at a lot of plants to get these recombination spots. These were also selected for variants, so there's a little bit of a give and take there. But we can't deconvolute at this point whether or not it's alt 4 or alt 3. It turns out alt 3 is also in the same region. So there's 2 alt genes that it actually could be. And this is important because when you look globally at the number of genes in the cannabinoid pathway, what you see is that we have the 2 main places that we get a lot of variation in the copy number of these genes are in BKR and alt4. And we do get a lot of variation in CBC, acid synthase, but really the other ones have limited variation. The way that I interpret this is that there's a massive potential still in the fatty acid pathway leading up to both the terpene and cannabinoid pathway. And so there's a lot of work that we can be doing on this pathway. The plant is already doing it, and this is probably a small snapshot of the potential of the plant, because our germplasm, even though we covered a large swath of germplasm, there's still unknown genetics out there. So BKR and all 4, the variation at them suggests that there's still a lot we can do. All right, so what use is a pangenome if you can't access it? So our main goal is we want to make sure that everybody has access to this. We've created a website that you can go to on my webpage. If you do Todd and Michael at Salk, you can find it. We have all these tools that you can use. You can basically do open up a genome browser. We have this tool that I like to use called Ortho Browser, which basically allows you to do multiple sequence alignment of any gene that you want to look at across the pangenome. There are other plant pangenomes there if you want to look at them. If you just type in Cannabis Pangenome, there's a whole bunch of resources online. Please check that out. All right, so what did I tell you? The main thing is there's a beautiful reference genome in terms of EH23. We don't even talk about reference genomes anymore. We talk about anchor genomes. The pangenome we think of as the reference now. But if you wanted one genome to look at, I would suggest that one. The pangenome is made up of lines from USDA. This means that they would be available for you to pair genomics with actually having plants. Turns out that cannabis is highly variable across at least the genomes that we've looked at. And this is mostly due to tandem duplications. There hasn't been a whole genome duplication event in quite a while in cannabis, and there's a lot of variation at the protein level. There isn't much variation in the CBD and THC synthase clusters. It could be that there's other variation out there that we need to access. And then finally, there is a bunch of variation in the upstream part of the fatty acid synthase pathway that goes into the cannabinoid, flavin, and also the terpene pathway. I think this is a great place to focus for research. A lot of people to thank. I would really like to give a shout out to Oregon CBD. This wouldn't have been possible without them. Obviously, I didn't do a lot of this work. I just get to talk about it. My lab is wonderful, and thank you.